Okay, thank you, Miss Jenny. Okay, so yeah, good morning, everyone. I'm Kitley. Um, I'm the field application specialist from Apical Scientific, and if you do contact with support from Apical, then I believe you know me. Okay, yeah. Okay, so today we are going to share on how to maximize your success of single DNA sequencing with Apical. So for today's webinar, we'll have two parts. In part one, we will talk about what is single DNA sequencing, and then also a little bit on the workflow of single DNA sequencing and how to prepare the good template for your sequencing, which will ensure you to get a more confidence result. And also, I will go through a little bit on the data analysis part. Okay, and then after that, we will have a 5 minutes break and come back for the part two, which is the exciting part, where we're going to introduce to you our new and very much easy to use online ordering system, the AO2. Okay, yeah, so without delay, let's start on one. Okay, so what is single sequencing? I believe everyone know about PCR, right? So single sequencing is something that is similar with PCR, but yet it's different. Okay, so what is the different? Actually, in the basic concept, sequencing and PCR, they are about the same. They have the primer. They are using the NTP, the DNA polymerase in the reaction. Okay, and also you will put this reaction into the thermocycler to perform to run. Okay, but the difference is in PCR we have the NTP. Okay, so this one. So in PCR we have the NTP. The NTP, it has a 3' hydroxyl group in the base that allowed the formation of the covalent bond with the next coming nucleotide. So while in sequencing, we actually have something that we call DDNTP. So this DDNTP, it will carry a H hydrogen group at the 3' end, which it will stop all the possible formation, covalent formation with the any coming nucleotide. Okay, so for this one, we actually call it as the strand termination chemistry. So in sequencing, we are using strand termination chemistry, and also there will be the NTP inside. So sequencing is a linear reaction where, for example, when you have 10 DNA, then there will be around 10 extended fragment found in the first cycle, and then and also the cycle is limited. So let's say if you have 40 cycle, then you may have about 400 extended fragment, something like that. So that is it means the read length may be up to 400 or less. Okay, but in PCR, it's an exponential reaction. So because PCR is using two primer and there is no strand termination chemistry inside the reaction, so PCR up up to the end, you will get um many copies of your target DNA. So this is the difference of PCR and sequencing. And also one more thing is in sequencing, because we are using one primer. Okay, so usually the primer efficiency of the primer will be very much important. So sometimes when you find that your primer can give you PCR product, then why it doesn't give you in any sequencing results, or it give you a very short result, something like that, is because the priming efficiency which is different. Okay, so that is the difference of PCR and sequencing. Okay, So, what is the workflow for sequencing? In sequencing, first you will need to make sure your DNA template is clean. Okay, so for PCR product, you may need to do some purification to um, get rid of all the DNA polymerase, um, all the excess primer, so that your PCR product is a clean double-stranded DNA. Okay, so what about plasmid sample? Do you need to perform any purification for your plasmid sample? The answer is actually no, because plasmid, when you perform the plasmid isolation from the host cell, is actually it actually means that you have take out the pure circular double-stranded DNA, which is the plasmid from the host. Okay, so the plasmid should be pure when you isolate by using commercial kit. Okay, yeah, so for plasmid, there is no need to perform any purification. And then after determine the pure template, then we will quantify, and then we'll know how much of the DNA template that should be used in a cycle, in the sequencing cycle. Then we will sorry, then we will add in the chemistry, okay, strand termination chemistry, where we actually using the big dye terminator, and then put in the forward or reverse primer in different reaction, and then we run it in the thermocycler. Okay, so after the cycle sequencing, then we will perform a post-sequencing purification. Okay, so this post-sequencing purification, the important is to remove any excess of the dye, because this chemistry, this excess dye from the DDNTP, they will later on affect the signal determined when we run the sample in the capillary electrophoresis. Okay, yeah, so we need to perform a post sequencing purification. Okay, then after that, the after determine the clean DNA, then we will dissolve all the extended fragment from the sequencing cycle with high diformamide and load into the genetic analyzer. So the signal come out from the genetic analyzer is the raw data. So it's not that easy to actually analyze it. So you need a software which we call this as a base calling. So we need a base caller software to analyze the data and send the result to you. So from there, you will get your result in clean electropherogram, something like that. So that is the workflow for Sanger DNA sequencing. Okay. Okay, so this is a little bit on the info on our DNA sequencing service. We are using high throughput sequencer. So the model is ABI genetic analyzer model 3730XL. Okay, and then there is 6 main service that we have. So a single pass sequencing is for purified PCR product, and also plasmid sample that you send in tube, then it's under a single pass sequencing. So if you want to send in the sample in 96 well plate, yes, you can. So you can just order for high throughput single pass sequencing. And if you maybe you have you don't have any purification kit for your PCR product, then you want to send us unpurified PCR product, yes, you can also. So it is under the DNA sequencing service plus service. Okay, so this service, if your sample is showing one band, then we will perform the purification, PCR product purification directly for you. But if your sample is showing two band, then you may need to help us to cut the gel, okay, cut the desired band and send the gel format to us without any solution inside. And then also we have difficult template sequencing, which this is suitable for template that is high in GC, that is maybe have homopolymer region, something like a long repeat of sequence like TTTTT, something like that. Okay, so this is for difficult template. And I would say that still sequencing, they have some limitation on all this difficult sequence. So if the difficulty of the sequence, um, it can still be passed through in by using this difficult template, then it's okay. But if it cannot be passed through sometimes because there is limitation in single sequencing, then you may need to think of other method. Okay, so this one, if you're facing anything, you can contact me. I can advise you on that. Okay. And yeah, also we have a ready to load sequencing service. This is customer need to prepare everything, and we just help you to run in the genetic analyzer. Okay. Okay. So for our sequencing service also, we have a wide range of free universal primer. If you are using any universal primer, for example, like 16S universal primer, we have all the 27F, 1492R, everything. Okay. And also we 100% quantify your DNA, which is important because every sequencing reaction, they will kind of very depending on the DNA amount that present in that reaction. So it determine how um what is the read length that you will going to have later on. Okay. Yeah. And the rest I won't go through it too much. Okay. So later on, if you're interested, you can go to our website to have a look. Okay. Okay, so we go into how to prepare a good DNA template for your sequencing, and why is it important? Okay, okay, because a good quality of DNA template, it actually means a clean the clean and pure template. Okay, so clean from inhibitor or any contaminant, so that this will increase the possibility for you to get a satisfied result. And it should also have a good concentration, which ensure sufficient DNA that present in the sequencing reaction, which it will increase the signal and the read length for your sequencing results. Okay, so how to get a clean and pure DNA template with sufficient DNA? Certainly, first you need to know what is the contaminant, so only you can get rid of all these contaminant, right? So you need to know the potential contaminant in your sample. For PCR product, the most common um, contaminant that you can find in PCR product, for invisible one, it may be like organic chemistry, chemical like the salt from the buffer, any excess PCR primer. Excess PCR primer, they will give you noise in your result. Okay, so like excess DNTP enzyme, all this is the contaminant that possible to inhibit your sequencing. Okay, and the invisible contaminant, something like you will see it when you run in the gel. It possible like this one is the primer dimer, or any unspecific band or smearing that is it means presence of short DNA that is inside your sample. So all these, they may causing background noise to your sequencing. Okay, so this is the thing that you want to get rid of it before you can use your sample for sequencing. Okay. Okay, how about plasmid? As I mentioned just now, plasmid, they should be pure, right? But sometimes after plasmid extraction, if you use too much of your um, um, cell, then you may possible to have protein contaminant. You still possible to have RNA or chromosomal DNA inside your plasmid. Okay, so that is all refer to the plasmid extraction upon the handling itself. Okay, and also some contaminant possible like organic chemical like phenol chloroform. If you are using a conventional method to extract, then that is also possible like detergent. All this is the leftover from plasmid extraction. Okay, so for plasmid is much more easy. You just need to use a commercial kit that is um, good, and then you just follow the protocol. Then it should able to give you a clean plasmid. Okay. Okay. So to get a clean and pure DNA, you need to eliminate all the potential contaminant. So how you perform a PCR product purification? There is a few method that you can use. Um, the most common one is the column clean up or magnetic beads purification that can be used when you find your PCR product actually is showing single band in the agarose gel quantification. Okay, so but if you found that there is more than one band in your gel after you run the PCR and then the gel quantification, you find there is more than one band, then you should go for gel extraction purification. You need to cut out the specific band, the target band, to do the purification. But sometimes that is possible of to get a very close band, then it may be difficult for you to perform gel extraction purification. There is case like this. Okay, so that is possible. Okay, so if you find something like this, you need to discuss, then please do not hesitate to contact us. Okay, and as for exocept, exocept is actually a type of enzyme purification, which it actually using the exonuclease will the exonuclease will remove the excess primer, while the stream alkaline phosphatase will remove the remaining DNTP. But this method, they do not remove any salt and enzyme that is present in PCR. So the purification may not actually complete when you use this exocept purification method. Okay, and the DNA purity is sometimes they may be a question if you're using this exocept. So according to our experience, there is some customer using exocept method, and there is they it actually affect the sequencing result. It may give you a shorter read line. You still can get result, but it will give you the shorter read length. Okay, for plasmid sample, the important thing to remember is to select single colony. So to avoid any satellite colony. Satellite colony, they may form when you over incubate your cloning plate. Okay, so that is possible. So try to avoid any over incubation, so avoid any satellite colony and make sure you pick the single colony for plasmid extraction. Okay, and for conventional method, which means you will perform, you will prepare the lysis buffer yourself. So for that, we will advise try to avoid to use conventional method and try to use a commercial kit to extract your plasmid. Okay, because commercial kit, they will come in a more complete, and the lysis buffer, they are also more consistent. Okay, so yeah, try to use a commercial kit for your plasmid extraction. Okay, and one thing to take note, okay, some of the elution buffer in commercial kit, whether for PCR product purification or extraction, um, they may contain EDTA, and EDTA is actually a potential inhibitor for sequencing. So we highly recommend you to elute the DNA in DDH2O for sequencing. If you are going to send your sample for sequencing, do elute it in the DDH2O. Okay? Okay, so now you get your clean and pure DNA. How about the DNA amount? So how you make sure the DNA amount in your sample is sufficient for sequencing? So you need to check the DNA amount. You can use agarose gel electrophoresis or using a nano drop or spectrophotometer. If you're going to choose one, okay, we will highly recommend you to use the agarose gel electrophoresis. Why? Okay, because nano drop is actually also a spectrophotometer, so it's actually using a light absorbent to give you the reading. And the readings sometimes they will be higher. And while you using agarose gel, because agarose gel it is using a DNA stain, so DNA stain they will more specific to detect DNA rather than other things inside your sample. Okay, so it will give you a more accurate result. And the the possible for agarose gel is you may have possible to um estimate the intensity of the DNA band that is generated. Okay, so for this one, actually you can always compare the intensity of your DNA, the sample, with the DNA marker that you're using. So do run your agarose gel together with DNA marker that you know what is the concentration of each of the band from the DNA marker. So from there, you can estimate and the amount in your sample. Okay, the DNA amount in your sample. And also for gel, we always recommend our customer to run by using just 1 microliter of your sample. Why 1 microliter? Why you cannot use 10 microliter, 5 microliter, or even more? Because um, you actually need to estimate the DNA amount per microliter. It's the concentration of your DNA, of your sample. It's the important. Okay, because in single sequencing, each of the reaction they actually have the limit of the volume that can be used in the sequencing reaction. So we cannot put too much. So um, maybe when you load 10 microliter of your sample into a gel and you see a very bright band, yeah, when you use 10 microliter, right? So, but in sequencing, we cannot use 10 microliter. So how? Yeah, that is why we recommend to use 1 microliter. So from there, you can estimate the concentration of your sample. Okay, yeah. So this is just to share with you, and uh from our advice, okay, from our experience, um if you want to run a gel for a big product size, you can use 1% agarose gel, and a small product size you can use 2% agarose gel. That is to increase the resolution of the gel. Okay, yeah. Okay, so once your sample is ready, you may actually store them in 4 degree before shipping. So or before we go to collect your sample. Okay, but this is suitable if you are going to store the sample for a couple of days, like in within 1 week, something like that. Okay, so but if you're going to store your sample for more than 1 week, then it's better to store at -20 until shipment. Okay, and DNA, they are actually safe to ship in room temperature. It's around 1 to 2 weeks. They are safe in room temperature. Okay, the important is there is no DNA's contamination in your sample. Then the DNA will be safe and nice when it reach us. Okay, even though it's in room temperature. Okay. Okay. Next is after you send for sequencing, you'll get your result, right? Okay, so how to read the sequencing results? When you receive our sequencing result in the zip file, so you need to extract all the file out, and then you will find that there will be two types of file for each of the reaction. Okay, so um, each of the reaction there will be a AB1 file. Okay, so this AB1 file, it need to be opened with a specific software. So we recommend our customer to use sequence scanner software because it's from ABI, which we actually using this analyzer, genetic analyzer. Okay, and then another file is the SEQ file. This SEQ file is you can open with a notepad. Okay, and SEQ file is just the the extracted all the sequence from the AB1 file. So the important things is to look into the AB1 file and not to the SEQ file. The reason why? Because in SEQ file, when you open in AB1 file, when you open with sequence scanner software, you will find the quality value bar. This quality value bar. So quality value bar, it will show you in three color. So here you can see that it's three color, right? The blue, the red, and the yellow. Okay, so these three colors, they actually refer to the confidence level of the base caller to call the specific base. Okay, so it means for example, like for red bar, it actually means the QV, the quality value is lower than 20. Okay, so lower than 20, it means the software call this, but the the confidence level is very low. Okay, and for QV20, it will show you in yellow bar. Okay, this is also the probability for call wrong call. Okay, so for QV more than 40, they will be in blue bar. Okay, so this is the one that we want to have fit in our result. Okay, so you want a blue bar to show in your result because it actually refer to a 99.99% the call is correct. So the confidence level for this base caller to call for your base, this specific base is very high. Okay, so this is something that you want in your result. Okay, so all the quality value of the base caller, they are actually determined by the base calling software, and it will show you the confidence level. And when the quality value is more than 20, the probability for error is 1 in 100. But when it's more than 40, the probability of error is 0.01% only. Okay, so this is the high confidence level of the base calling. Okay, okay. The important for base calling, it actually will help you to know which sequence that you can trust, which base that you can trust, and then you can choose them for further analysis. You need to choose all this trustable base to use for your result analysis, for alignment, for blast later on. Okay, so all this is the base calling is very important. And also you can determine your SNP. If you are studying on SNP, then you can determine the SNP very easily from the result. Okay. Okay, so if you look at the report, you get the report from the sequence scanner software. You can click at the report, then you will see that there is a CRL, so contiguous read length. It is actually the uninterrupted stretch of the high quality base call. So usually we will call it as the longest region that have blue QV bar. Okay, so the CRL is the one that you want to refer to later on for your result analysis. Okay. Okay, and what is trace score? Trace score is the average of the quality value inside this CRL. So as long as it is above 40, then it means this CRL is good to use. So you can use the sequence inside this CRL for an alignment or blast later on. Okay. Okay. So what is good result? Okay, good result. They should have the clean space, the um space between the peak, they should be even, and it will show you sharp peak. Okay, then there should be no background noise or very little or small background noise. They are acceptable as long as the quality value, this quality value show you in blue. Okay, then if your sample size is shorter than 900, then you should find a stop point at the back of your sequencing result. Okay, yeah. And then yeah, just to show you, this is the raw data that you can see in raw data. They should have a clean and nice baseline in your raw data. And raw data, if for short fragment, it will actually stop at A here. Okay, so as long as the baseline is not flying up, then it means your DNA sample is actually pure. Sometimes you will see that there is some flying up and a white region here, and then the baseline flying up. That is actually your sample have some impurity. Okay, yeah. Okay, then in sequencing result, it is common that you will see some low signal in the front part, because as what I mentioned just now, you after the cycle sequencing, we will perform a post sequencing purification. So this post sequencing purification, it will causing some loss of some short fragment. So this is the loss from the short fragment. So usually it won't be very serious. So it can be up to like maybe less than 40 base, that is noise, something like this. So this fragment is after the priming site, then only it start to have this loss. So that's why in sequencing result, when you get your sequencing result, you'll find that you cannot, for example, the sequencing result is the reaction is using forward primer, then you will find that you cannot see the forward priming sequence, because the primer need to bind, then only the sequencing will start. So also you will have some loss in the front part after the loss of some sequencing sequence after the priming site. Then only you start to get a good read from there. Yeah, okay, so this is refer to a good trace, good sequencing result. Okay, so there is other sequencing possible of result that you will get. So this one is for weak signal. So weak signal, you can find that actually there is undetermined baseline. So you cannot see any baseline. You cannot recognize the baseline from the raw data. Okay, and the electrophoregram, it will not able to show you a very clean and sharp peak. Okay, so you need to refer to all this to analyze your result. Okay, so usually this is due to possible there is too less of DNA template in the sequencing cycle, or too less of the primer in the sequencing cycle. Then it may causing weak signal. And also there is possible like there is present of any contaminant or inhibitor in the reaction that will inhibit the sequencing. Okay, yeah, so you have very weak signal that is generated. Okay, okay. Another one is early signal loss result. So in early signal loss, you will actually see that the signal drop very, very fast in the raw data. Okay, so the electrophoregram, they will show you noise that is start uh, early. So if your fragment is long, for example, like this result, if your fragment is long, you should actually get a good read up to around 900 and above. But now it actually showing you the signal start to noise start from here. Okay, so it's a early signal loss result. Okay, and this can sometimes because of insufficient of DNA in the cycle, or incorrect sample primer ratio. That's why we need to quantify your sample before the sequencing reaction. Also, another possible is that can be also inhibitor. That's why use giving us a pure and clean DNA is very important to get a good result. Okay, so another one is erupt signal loss. Erupt signal loss, it doesn't mean your sample have impurity, or it also doesn't mean it's not enough DNA. Erupt signal loss result, usually it happen in plasmid sample. Okay, why? Because plasmid samples sometimes they are easily to form secondary structure. So all this secondary structure, when it actually happen in for example like this region, then the polymerase, when it is in the sequencing reaction, the polymerase may not able to pass through the secondary structure, so it will drop off. Okay, so yeah, this is the where you find that the signal will drop from upon the sequencing. Okay, so you will find a very sharp drop in the signal. Okay, so this is erupt signal loss. Okay, then there is actually a lot of different type of result that you may receive. You may also receive some data that is noise after a specific point. It can be possible of insertion or deletion happen in your sample, because it possible your sample is a heterozygote, something like that. So there is many possible result that can happen. Okay, so you can go to our website to take a look. Okay, yeah, so you can find this in our resource FAQ that site. Okay, and if you need more information of our sequencing service, for example, you need to know what is the protocol that we are using, what is the machine that we are using, what is the chemistry that you are using, so that you need all this information to write in your report, in your thesis, in your journal, right? So you can always go to our website to take a look on it. Okay, yeah. Okay, so that's the end of my part one, and we'll take a break from here. So do come back for part two. Okay, so we'll take a break. Maybe we can come back at 10 or 10:50. We can come back in 10:50, a few minutes time. Okay, yeah, see you. Do come back. uh, okay, yeah, we have some question here. Maybe I can answer the question first. Recommends to use the sequencing primer as amplification primer. Example, sequence for 27F1492R primer. It's slightly different from the original paper. Okay, you can, if you're using the amplified primer, the primer used for PCR for your sequencing. So if you're going to use to design your primer for PCR and then later on to use in sequencing, there is some additional rules that you need to add on your primer design. So for example, you cannot have like TTT at the end of the primer that will affect your sequencing later on. It may give you some noise. Okay, and then if, for example, if you're using the 27F and 1492R primer, for sequencing, if you find that the sequence of our 27F is different with the sequence that you use for your PCR, I would advise, do send your primer for sequencing rather than use our 27F. Okay, yeah, there is some different in sequence from from paper to paper. Even though it's 27F, there is some different in sequence also in between papers. Okay, so you need to double check. So you can find our primer, our universal primer sequence in our website. Do confirm the sequence is the same with your sequence, the sequence that you use for your PCR. Then you use our primer for sequencing. Okay, yeah. Another one. What is for the recording? Any other question actually about the sequencing? Still okay. Actually, about the sequencing, is single sequencing still prevalently used rather than next generation sequencing because of the limitation of single sequencing? Okay, single sequencing, I believe single sequencing and next generation sequencing, they also have the limitation. Okay, yeah, so the limit for single DNA sequencing, I would say that if you have multiple sample inside, because single sequencing, you can only sequence for one type of DNA. So if you have multiple sequence inside, then you cannot use single sequencing. Then you need to go for next generation. And also single sequencing, it may take up more longer time if you want to sequence like a huge DNA. So like if you want to sequence the whole genome, then it will take up very long period to perform all this. So for next generation, you can um, make it fast. But if is it really need to go up to next generation sequencing? Because or sometimes maybe you just need to have a look on your PCR a little bit on just to verify the sequence, or you want to, like for example, you want to check your qPCR product sequence, then you just send for sequencing. You won't use the next generation sequencing. Okay, so there is pro and con. So you need to know what is your objective. Okay, yeah. I have a question. What is the advantage of sequencing to PCR? You mean okay, sequencing, it's actually you need to get the what is the sequence in your PCR product. If you after you run a PCR, you get a sequence from your target gene. Then after that you run a gel, so you know the size only. You won't know what is the sequence inside. So for sequencing, you send it for sequencing to know what is the sequence. Okay, yeah. Raise your hand. Oh yeah, hi, Abu Bakar. Do you want to open your mic?
Hello, thank you so much.
Yes.
We are really grateful for the good presentation we're having today from an expert like you. Having said
Thank
that,
you.
please, I have a single question about SNPs, single nucleotide polymorphisms.
Yes, okay.
I just received my sequential result within last week, and that is one of the things I want to do. Currently, I have two different set of microorganisms whose sequences need to be aligned to compare to know the regions of the polymorphisms, if any. Please, I want an expert advice about that, even also including the software to be used in your, yeah, please. The software to use and how to go about it, please. I really appreciate it.
Yeah, as mentioned just now, for sequencing results, we recommend to open the AB1 file. So when you open the AB1 file with our recommended software, the sequence scanner software, then from there you can easily detect the SNP actually. So you just SNP, it will actually show you in double peak, and you will find that the sequence beside it, it should be a good sequence. They will be in blue QV, and then it will have a peak that is showing double peak. Then that will be the SNP that you want to refer to. Okay, I hope I answer your question.
Yes, so thank you so much. I really appreciate it.
If you still need me to like show you, or where is the SNP, perhaps you can send me an email at support@apicalscientific.com. Then I can print screen your result and show you where is the SNP. I hope that will
Okay.
help.
Yes,
Yeah. Okay.
I will do that.
Okay.
Yes, ma. Thank you.
Okay. Thank you. Okay, Liao Ling. Okay, I would like to ask if my sample is in gel form, what temperatures should I store prior? Yeah, you can store it at 4 degree, and for gel format, I would say try to avoid a long storage because still gel, it will still easily to dry up, and when it reach us, we may have some problem to really dissolve all the gel. So try to avoid um, a very long storage or more than 1 week or 2 weeks. Okay, yeah, try to avoid that. Any advice if we receive a weak signal from the sequencing? Weak signal, there is many possibility as what I mentioned just now. There is like your DNA is not enough, maybe insufficient. Then certainly you will get a weak signal, and that is possible like inhibitor or your primer amount is not enough. So there is many possible reason. Okay, so for this one, you need to further check. Sometime if you maybe if you are going, you have two reaction, then yeah, just try to read from another reaction. Okay, yeah. I think we will need to stop the Q&A section first, because as I mentioned just now, we have a very exciting topic to share with you. Okay, so I will pass this to um, Fifi, Miss Fifi, to show you the what is exciting from us. Okay, yeah. Then later on we come back with the question. Okay,
All
to
right.
you, Fifi.
Right. Thank you, Kelly. Thank you, Jenny. So, I'm Hafifi. So, I'm the sales support specialist for Apical Scientific lah. So, today I will be introduced to you our exciting part, which is the Apical Online Ordering, or in short, we call it as AO2 system. All right. So, so the contents of this presentation will be an introduction on the AO2 system, and also the some key features that is available in this AO2, and then some important announcement to our dearest customer and also distributor, international distributor. And then the last part I'll be covering on some live tutorial on how to register and also on how to place DNA sequencing ordering in this AO2 system. All right, so we move on to our AO2. So welcome to our Apical Online Ordering. So this is our interface. If you ever log in or visit to this AO2, which is the URL here, http://order apicalscientific.com. So this AO2 is actually replacing our current 100 system, our old online system, and also manual Excel order form. All right. So we have officially fully implementing this system since its launch in September 2023. And then we have actually receiving many orders from many different customers, and from local and also international, from international for services such as Sanger sequencing and also DNA barcoding and avian DNA testing lah. So for now, we are still waiting other customer to fully utilize our AO2 starting from today. So yes, that's why I'm here to introduce to you this AO2. All right, so these are some of the key features of our AO2. So yes, you can order at your own convenience, 24 hours, anywhere and anytime. And then it has this tailored account management where you can easily view your order history, your order results, and also your prepaid account with us, which is SOID. So some of our principal or PI PI will have this SOID account with us, right? So your email will be linked into our SOID account, and then you will be able to see your account balance afterwards. And then we have this responsive support from our lines of dedicated customer care. So you can have you can just email us or WhatsApp or live chat to get a prompt response from us lah. and then your data is secure with us, means your order credential and all your result will remain confidential with Apical Scientific. So that is our promise. And then we also have this data sharing made easy, so you can actually download and also easily share to your DNA sequencing result to your research groups. And then high accuracy. So when you place online ordering with us, you can simplify the data input and also minimizing the human error, especially from your side and also our side in capturing your orders. Okay, so this is why we need to move forward to the using of online ordering. And then the cloud storage, you can get your results or order info wherever and where and whenever you need, as long as you have internet coverage. Okay. Next, we move on to an important announcement. So this is the important announcement. So I hope you guys can take notes or take some what we call some attention on this. So we will stop accepting 100 orders, which is our old online version, and also manual order form, which is the Excel format, starting from 1st of December 2023, which means there will be no more 100 orders. And also maybe we, as mentioned by our CC, they will also receive a manual order form, but in slower rate. The processing will be in slower rate. Okay, so yeah, I have to stop slowly. Thank you, Jenny, for reminding. All right, so the manual order form you can actually send us, but we will be less prioritize the ordering process lah. So we will prioritize the AO2 online ordering first, then only we'll go to the manual order form. So to smoothen the orders and also to make sure that there is no delays on your orders, please start utilizing the AO2 from today. All right. And then the result retention at all online ordering, which is our old portal 100, the Biz Asia. The result will only be available for download until 31st of December 2023, which means you are advised to download the results, all your results, and retain a copy into your physical hard drive or your PCs before the deadline. So make sure that you download all the result, yeah, before the deadline. Okay, so for any special request on data retrieving after this deadline, you may email to our sequencing@apicalscientific.com, but the processing time will take around 1 week, as our team will need to retrieve the data in our database lah. It will take quite long time. So please download the result before the 31st of December 2023. And then the result retention on this new AO2 will be stored in the new online system for a maximum of 24 months or 2 years only. So yes, please download the results for this AO2 and then save it into your physical hard drive lah. So any special request on data retrieving, you can just email to us, same as what I told before lah. And then for this AO2, it is advisable to our user to use an individual email to register the account and to place the order, not using lab or any general email. This is because all incomplete order information and also all email regarding your orders will be sent to the registered or unique email only. So to smoothen the process, to not delaying your order, make sure that you use your individual email instead of info@blah.com. So please use your individual email. All right. And then, okay, so this is the most the most exciting part, which I'll be showing you the live tutorial on how to account setup, to do this account setup, and also how to place an order through the AO2 lah. All right. So for our existing loyal customer with 100, the active user, you can simply log in into your AO2 using your email, the registered email in 100, and then set up a new account lah. So I'll show you. All right, maybe I can stop presenting this here for a while and show you my screen. A moment. A moment. Okay, so you guys may see my screen now, right? Okay, so this is our interface of AO2. So this is the URL, order.apicalscientific.com. So I'm giving a tour, a live tour for you guys here today. So okay, what is the service that is available, what we offer to, or what we offer to our customer to order in this AO2 is, the first one is services under standard sequencing, which is single pass DNA sequencing for purified PCR product and plasmid. And then we have like high throughput for submission in plates, and then ready to load sequencing. And then we have the sequencing plus as well for unpurified PCR product, direct colony sequencing, primer walking, and some MBS services, which is DNA barcoding, and also avian DNA testing for bird sexing. So these are the services that we offer under this new platform. All right, so we'll show. Okay, for one, as I mentioned before, for 100 existing user, you may just click forgot password here. Sorry. Okay, I need to log out first, sorry. And okay, log in. Okay, you can just click forgot password here for our 100 user, enter your email address linked to the 100 email, and then click submit. Afterwards, you can actually you will receive an email with a temporary password lah. So you may actually log in into your this AO2 using that your email, and also the the temporary password. Okay, so that is as easy as ABC, right, for the 100 or existing customer. And then for our new user, so I can show you on how to set up a new account with us lah. So okay, so this is the, for new customer, you can enter your email, your first name, your last name, uh your telephone number, date of birth, and this one, the most important thing is the country selections. Okay, since we have like two groups of customer here, our dearest international customer and also dearest local Malaysia customer, so you can choose the country from the drop down list here lah. So if you are from Indonesia, you can click Indonesia, and then click your distributor, which is PT Genetica Science Indonesia. And then for Malaysia customer, here Malaysia, so the distributor will be Apical Scientific Sdn Bhd. So if your country is not available in this drop down list, you can just choose countries other than Malaysia, and the distributor will be tagged to Apical Scientific Sdn Bhd. Okay, I hope you guys are clear on that, because this determination of countries is rather important for us to link you to the correct price group. So please choose your correct country and also correct distributor. All right, and then how did you hear about us? Okay, this one just okay, maybe like FB, you can just enter like that. And then I'm not a robot. And then please check this term and conditions on our AO2, on the services and also on the products. So afterward, you can just click create my account. All right, so after you create a successful create of my account, your account, you can receive an email from Apical Scientific saying that you have successfully registered with AO2. Okay, so that is how you set up a new account with AO2. And then we move on to how to place sequencing orders using this AO2. Okay, since today we'll be having like two groups, international and also local customer. So I'll be segmentize this ordering process into two groups lah. The first one will be ordering single pass DNA sequencing for local customer, and sequencing plus for our international customers. All right. Okay, so okay, I need to log into my account first. Moment. Okay. All right. So in this account, okay, after your successful login into your account, you have to fill in your details credentials lah, such as your addresses, and then change, maybe you want to change your details. In here, you can change your detail, but email you cannot change. It is unique to your own individual registered email. Okay, so addresses is important so that you can see where to collect the sample letter and whatnot. All right, and then here we have like reward points. So the reward points is for any purchase that you have completed. Yes, you will be subjected to certain rewards amount. And then the store credits, this is where you can actually link to the your SOID balance lah. So once you complete one transaction in this AO2 new system, you will be able to see this balance in the the store credits here. So only one email will be linked to this account lah. Usually your principal investigator or your maybe your supervisors lah. All right. And then we move on to how to order the single pass DNA sequencing, which is for the purified PCR product or for plasmid, purified plasmid. All right, so this is for local customer, yeah. So we can just choose the type of sample here. So for purified PCR product, and then order submission is by using cubes. So and then you can choose any add-on DNA template here. So as per the name, add-on. So there will be an add-on charges as well for these services lah. So it will all comes with charges. This add-on DNA template, add-on reaction, add-on primers, and also add-on orders. So this will all come with add-on service charges. All right. And then for the most important parts in this ordering part is the where your sample name or sample credential or your order credential will be. So if you can see here the red fonts on standard sequencing, click onto this standard sequencing, and then there is an order form download lah. So the order form will be looks like this. So this is the I fill in this form pre-fill. All right, so they will be having like three tabs, different tabs. So important for you to check on the information tabs. So read the information here, then only you fill in the order form. Okay, so in this ordering form, you must not use any.
You can
Sorry?
see the Excel file. We can't see the Excel file.
Oh, you can see. All right, sorry, moment. Okay, all right. Is it clear now? All right, I think it's clear now, right? Okay.
Maybe
You can't see?
Yeah.
Oh,
maybe
sorry.
You need to unshare and share back your screen. Thank you.
All right. All right. So now you guys can see the Excel form.
Yep.
Yes.
All right.
Yes.
Okay.
Yes.
Thank you. All right. So for this information tab, read clearly. And okay, so for this ordering form, you must not put any character, symbol, or number. Any special character or symbol lah, except space and also hyphen, which is the minus sign, and also underscore is accepted. Okay, only put alphanumerics, space, hyphen, and also underscore is accepted. Okay, if not, our system will not able to capture, and you have to cancel the order and remake the new order in this AO2. Okay, I hope you guys clear on the naming of the sample and primer name. And then the default unit sample size will be in BP. The sample concentration must be in nanogram per microliter, and primer concentration will be in micromole, and primer annealing temperature will be in degrees Celsius lah. So you don't need to put this in this unit size, because we refill it for you. And then, okay, if you are wishing to use a universal primer, you can check on our list of universal primer provided here lah. All right. So you don't need to send the primer. So you can just put the primer name instead in the ordering form letter. Okay. So for the universal primer, just put 0 on the primer concentration and also primer innerling temperature. So we know that you are using our universal primer. So don't need that info anymore. All right. And then now we go on to the order form. As I mentioned, there will be no special characters or other symbol except the underscore and also the hyphen and space is accepted in this sample naming and also primer naming. So make sure you comply to that instructions lah. All right. And then after that, you can save the order form. And then you may upload the order form in the section here. Okay, moment. All right. All right. So your sample name or your order credential will be previewed in this upload preview box. So you check that every credential and details is correct, then only you can proceed to proceed button. All right. So this is the address details. So as I mentioned earlier, after you successful login to your account, you must have to fill in your address details. Then only your address detail will be shown here lah. Or not, you can just put in another detail if you wish to have the other address to be registered in your account lah. All right. So I put this is just my dummy account. So this is just press to any. And then, okay, this is where it's actually differentiate between our local customer and also international customer, which is these two parts here, delivery and payment method, and also the complete order. All right. So for local customer, you'll be having this interface lah. So submission method and also the payment method is different. So for sample collection point, you may select any one of the sample collection points here. If your sample collection points is not listed, no worries. You can actually just choose any nearest collection point in this drop down list, and then we will just put any additional remarks here lah. Let's say level 2 parasitology something. Please collect, sorry. Something like that. Okay. It's actually our IT department is fixing the issue. We will put more collection point on this drop down list lah. So after this, you'll be, if your place is not available in here, maybe it will be available soon. So no worries on that. Just put any special remarks on the additional instruction box below lah. Okay, so in this additional instruction box as well, just make it simple. No need to write an essay. Make it short, simple, and meaningful. Because sometimes if you write like very long essay, our database could not capture the order lah. So make sure it will only just a simple message to us lah. For any additional instruction or special instruction to our CC or our production teams. Okay, and then next, if let's say you want to choose local courier, you can write down your AWB information here. And then if you choose to use in our reseller channel, you can just click the reseller channel. All right. And then for example, since I choose UM just now, so I can just click UM. Where is UM? Sorry, a moment. I can see UM. Okay lah. Anywhere lah. Okay, UMPJ. All right. So for choose a payment method, if you wish to pay in prepayment, you can just put your reference number here, or If you have purchase order number, you can just fill in your purchase order number here. Or if you have a prepaid account with us, you can just put your SOID account number here lah. All right. And then same as reseller, you can just put reseller if you wish to use our reseller channel. All right. And then you can click this terms and condition and click continue. So this is the final checkout page. Check that every information is true before you confirm your order. Okay. So I hope you guys are clear. Here, just confirm the order. All right. So afterward, you will receive the thank you email as well after you place the order. Or we call it as order acknowledgement email. You will receive an order acknowledgement email after you place an order with us. Okay. All right. So if let's say you wanted to check back your order, whether your order is true or not, the final checking before anything, can just click view here, click the order history here just now, order history, and then we'll come into this interface lah. View your order information. And then this is where you can actually see your order result, whether the result is ready or not, or the result is in progress, or there will be a status changing here lah. Possibly. All right. Just if you want to print the order form, you can also print the order form and attach it into the parcel after parcel or your sample ziplock lah. Okay. I think that is all for sequencing, single pass DNA sequencing. And then next is sequencing plus. So this is especially for our international customer. So for DNA sequencing plus is for if you wish to send us the unpurified PCR product or plasmid lah. So what you need to do is to type on the to choose the correct sample that you wish to send. For example, unpurified PCR product, and order submission is in tube or in 96 well plate. It's your call. And then choose any add-on DNA template or any add-on services that is available in the below sections lah. So as I mentioned, this will come with add-on service charges as well. All right. So you have like three options here. You may choose plate one if you plate with one primer, if you choose to send it in with one primer only per plate, or you want to choose two primers or the plate with two primers. So you can just click in here, and then also if let's say you want to submit it in tube form, so you click the tube form Excel format Excel form here. All right, so I will show you the okay, so maybe I can show you one plate format lah, 96 well plate. All right, so this is the plate one primer form will look. So yeah, as I mentioned, please read the information tab carefully. There is no special character or no symbol except space, hyphen, and underscore only will be accepted in this order form. And then the format of 96 well submission must be in the orientation of A1 until H12. So you must leave this 96, the what H12 the H12 well empty lah for our QC reactions. Okay, and then this is the list of universal primer. So you may choose from here, and you don't have to send the universal primer to us lah. All right, and then okay, so for plate format, please avoid to use same sample name. Okay, if let's say you have like same sample name, you must differentiate it into one for example, like this one, L1 PEPV 1 or L1 PEPV 2 or or anything lah. So make sure just differentiate between this the same sample name. Okay. So for primer name, since this one is only for one primer, so the same primer name will be provided here lah. Okay. And then primer concentration, since this is our primer universal primer, one of our universal primer, so the primer concentration and also primer annealing temperature will be 0. So you don't need to enter or any information on these two things lah. Okay. Then save this one and then you may upload the file. So now upload the file with the primer and then you may see the order preview from the box, preview box here. And then you can proceed. Okay, sorry because I'm using the local. So I need to log out. All right. So first, a moment. A moment for sequencing class. So I just repeat the same thing. The 96 file format and then upload the plate 1 primer. Okay, and then file will be uploaded here. Your order credential will be uploaded in the preview box below. And then you may choose, click to proceed. And then for this delivery and payment method, this is the address detail. So this, as I mentioned, this is for especially for our international customer. So from Indonesia, you can just click the, okay. So this is the where the interface is different lah between local and also our international customer. So if you choose to send your sample and primer to our distributor channel, then you can just click distributor and then choose a payment method also from our distributor, PT Genetika Sign Indonesia. And then if let's say you want to send us not through a distributor or direct through Apical Scientific, you may choose local courier and then just mention your AWB number here lah. Okay. And then for reseller, just choose reseller. So I'm going for distributor channel since this account is tagged into our distributor in PT Genetika. So any additional instruction here, if let's say you are having voucher, so just mention voucher redeem for certain number of voucher lah. Voucher XXXX, something like that. So this is one of the example of special instruction. And then you may click to the terms and condition. As I mentioned, just read the terms and condition before you proceed to any further process lah. Okay, click continue. And then you're able to see the summary of your order. Check that every detail and information is correct before you proceed with your online ordering. Okay, if let's say you're paying with prepayment, you can just upload your payment receipt here. This one. So choose any prepayment, your receipt. And then just confirm your order lah. So just the same thing, only 4 steps, and it's very, very straightforward and also customer centric lah. So you don't need to like wait for your distributor to approve your order, then only it will come to Apical Scientific now. So we have like cut those process. So every order from international customer will direct to Apical Scientific itself for smoother and also for what we call to shorten the ordering process lah. Okay, I think that is all. Okay, maybe like one important thing as well. would like to mention here. If let's say you are teaching your lab mates or anyone, so maybe you can just refer to our FAQ here. So we have like a set of questions for catering Sanger's DNA sequencing services and other services lah, especially for the ordering. Okay, so you may refer to our FAQ here. It is very useful for you as well. And if let's say you want to revise back on how to order, you can just go to setting up a new online account and contact guideline text here, and then scroll down a bit. You may find this how do I place an online order lah. So you can just click to Apical Ordering tutorial. You will be redirected to our YouTube channel. So you may see sets of video that have been uploaded. You can refer to this video as well lah, to recap on how to place online ordering with AO2. So I hope everyone is clear, or you have any questions on online ordering AO2? Question okay. So
there are some at the chat box.
Some at the chat box. Let me see.
And Jason already helped us to answer part of them.
Okay.
Maybe you can choose some that is important to.
Fifi, I think there is one question. Can we send gel and tube in one form course using the same primer? Using the same primer, but they want to send, the customer want to send it in gel and tube. It means different format. Yeah.
Oh, okay. I think this is a bit tricky. Jason, maybe you can help me. Jason?
Just to answer for that particular, we will advise you to submit two separate orders for that case, even though you're using the same primer, because of you're using different sample
type.
Yeah. So we will minimize the need on back end for us to change anything. So one order, one sample. Yep. Also to answer Aeon, the 100 could select the ready time. There's no timing feature in the new AO2 system anymore, so you only need to select the collection where do you want it to be collected, and we'll actually follow our schedule, our dispatch schedule to collect the sample. So generally, when you place the order, I believe the sample should be ready by that time. So we'll collect according to the dispatch schedule to when they're going and all those stuff. Yeah. So any changes or any further requests or anything, normally our customer care will contact you directly. In regards to sample collection, if there are any changes also, but generally nothing much should change from the current practice. If if the sample, what you have been practiced in 100 should almost apply the same in terms of the collection and all those stuff.
All right. Okay.
How to tell because they are using different primer besides in the universal primer list? I believe in the order form you can state the primer that you that you want to use differently, and then you can state another.
Maybe I can show you. Maybe I can show you here some of the information. So yes, in the primer name you can just fill in your primer names lah. If let's say you don't use any universal primer, you can just fill in your primer of interest in the primer box here. And another important things for this order form, the format is fixed. You must not change any information here. Okay, so if let's say you want to pandai-pandai make any another box here, it's not allowable. So our system cannot capture your order lah. So might as well we need to cancel your order, and you have to put a new online ordering with us. Okay, so please don't change any format on this order form. Please just use the format that we provided. Okay. I hope clear. And then any other question? How about the size of BV information? Same as the primer name, you can actually put on your sample size information in this column here. Just put your sample size. Okay, and then how can I cancel the order? I just noticed that I filled in the Excel form incorrectly and send the order already, but I do not send the sample yet. Okay, so how to cancel the order is you have to email us to sequencing@apicalscientific.com and saying that you will cancel this order ID, and I have made this order and place your new online order ID lah. So if let's say you have the order ID ready, then just mention your new order ID lah. Okay. No more question ready lah. Okay.
I think almost done. Or Jason, do you need to like highlight anything else that you wish to tell our customer?
I think the most important is, I guess, the announcement again that coming 1st December we will be stopping the online ordering.
Yeah. Right.
So that is the most critical part, because one of the reasons why we have this session is also to promote the awareness, so that come 1st of December when you are unable to place the order, you won't be trouble of his panic or say how come I can place the order, what's happening and all stuff. So we're trying to reach out as many people as we can to alert them on this. So yeah, if you have your lab mates or anyone that is actively submitting orders through 100, maybe you can pass the information to them as well, that you may start to actually one thing we will I would say advice is you start to use the AO2 now, so that if there's any issues, we can help you to troubleshoot it. But most of the I would say the common issues are mainly on the Excel form. When you upload the form, maybe you add in additional characters or certain additional columns. That's why we can't capture it. But I would say so far it's been quite easy to capture the orders. It's very straightforward. So
compare with 100, I think.
So give it a try. Let us know if you have any issues by emailing us or calling us. Yeah, that's it.
So
no. Okay, so means that if you are our existing customer, so please spread the words to your lab-mate, your friends, that we already migrate to a new system, which is easier for you to capture the order, and the registration also very straightforward. Once you register, then you pop straight away, you can get the password if you're a new user. So no matter you are old or you're a new user, so we are really hope you all can use our AO2. So remember this AO2 stand for Apical Online
Sorry, Jenny.
Ordering.
To answer Shamila's question.
Can.
Yes.
Yeah.
Postgraduate samples, if you want to send sample, they need to use their supervisor account. So this is what we advised earlier, because certain labs you have multiple personnel using. Sometimes in the previous practice you have multiple students using the same account. Well, technically, I will leave that this question to you guys if you want to continue the same practice. But generally, we would advise, if there's a new student or a different student wants to place an order, get them to register using their own email address, the one which the university provides or whatever not. And then under the payment section, you can put the supervisor's account details.
SOID.
SOID, so we know where to charge it to. Most of the time you have the SOID number, so when you place the order, just state the SOID number, then we will know who to charge to and all that stuff. We will have the records on our back end when we actually process the order. So yeah, I guess the important part is to utilize the one person, one account, one email, one account thing. Yep.
Okay, Thank you, Jason.